Welcome to the Dr. UUT channel. The topic is intrinsic activity. The lecture is available in Urdu. Let's proceed in English. What is intrinsic activity? When any drug, according to pharmacology, when any drug or medication or ligand binds to the receptor and that receptor sends the signal to the cell to show the action, that is called intrinsic activity or internal activity, shown by the ligand's stimulation of the receptor. The ligand will stimulate the receptor and then will show action inside the cell by means of the receptors. So what is the agonists and antagonists? Sometimes when these drugs or ligands attach to the receptor or bind to the receptor, the internal responses are shown, then they are given the name as agonists. Sometimes when they bind and to the receptor, then there is no any internal response shown, they are known as antagonists. These are the antagonists. Now there are following types of each: agonists and antagonists. Fully agonist. First we'll cover the agonist. When the medication or the drug binds to the receptor, it shows full response, internal response, or it shows fully intrinsic activity, then they are known as full agonists. And what are partial agonists? When they combine with the receptor or bind to the receptor with the receptor, then intrinsic action shown as usual is not like that. According to pharmacology, we say the internal signal will be more than 0 but less than 1. More than 0 but less than 1. That is called partial agonist. There will be action but not like full agonist actions. The word is inverse agonist. The name indicates inverse, opposite. How? Suppose this ligand is responsible to give the signal to this receptor, and this receptor will send the signal to inside the cell for the protein synthesis, suppose. In case of inverse agonists, now this is inverse. This will come and will bind with this receptor. Before, this receptor was having the function to show the action, but now this receptor will be stimulated oppositely. That now this, before it action was to produce the more protein, but now it will split or decompose the proteins. Suppose I'm giving an example. That's called inverse agonist. Meaning it will invert, it will convert into another style. It will go against the action that was specific for the receptor. So this agonist, which is going to do this kind of activity with the receptor, are known as inverse agonists. Then those agonists, okay? Antagonists, again, competitive, non-competitive, and functional antagonists. What are competitive agonists? When the medication or the ligands binds to the ligands, when they bind to the cell and they show internal signal. Generally, what is competitive antagonist? Here will come the antagonist. This is agonist. This antagonist will bind to the receptor, then internal signals before they were shown, now not. Because of what? Because agonists are kept aside because of competition. There is a competition between these two, agonists and antagonists. Now if you can overcome this process, means if I want to do the internal action, to promote the internal actions, what will I do? I will increase the agonists. Then the antagonists will disappear. The agonists will be free to bind to the receptor and to show the internal signal. Then the internal signals will be shown. Now we have got another that is called non-competitive. In this case, what will happen? We have irreversible and allosteric antagonists. Irreversible are when here is your antagonist and here is your agonist. Antagonist, if binds, this irreversible, I'm talking about irreversible one. Now if this irreversible antagonist binds to the receptors, then there will be no any kind of internal signal. As like competitive, if you increase the agonists, then the antagonists will take a side. But here in case of irreversible, as the name indicates, it is irreversible. This antagonist will covalently bind with the receptor. Then no internal signals will be shown or will be given to the cell to show actions. So then however you increase or decrease the concentration of agonist, nothing is going to happen. And allosteric. Now, in case of allosteric, what is the function? It has got a very different mechanism. Allosteric, what will it do? Allosteric will bind to the receptor from another side, from another receptor. So what will happen to, if they bind to another side of the receptor, not another receptor, from another side of the receptor? When allosteric bind, there will, however the case is with the agonist, whether agonist is binding or not, there will be no action shown by the internal actions will be not shown by the agonists. Why? Because allosteric has now got the side at the another side of the receptor. There, that's why the allosteric will cover that side. Then agonists, whether they are binding or not, then there will be no any kind of action shown, because these allosterics have got somehow greater effect or effective characteristics of antagonism. When they bind, then they will not allow the signals of the agonists to the cell, inside the cell. So this is the example of allosteric antagonists. And we have got here the functional antagonism. Now what is the mechanism of functional antagonism? Here the mechanism you will get by means of an example that you have got lungs, where here the smooth muscles of the lungs have got two types of receptors, histamine and epinephrine. Receptors are of beta 2 and H1. H1 is for S-histamine and beta 2 is for epinephrine. Now here, smooth muscle is a single muscle, okay? Here if you get a single muscle, smooth muscles of the lungs, when histamine they come, if histamine binds to the receptor of H1, it will cause the contraction. It will cause the contraction of the smooth muscle. Now what is functional antagonism? The receptors, when they activate, they will get the functionally antagonistic characteristics. The function of the receptors in a different way. Now another receptor is here for beta 2. Here, when epinephrine come, they will stimulate the beta 2 receptor, which will cause the relaxation of the smooth muscles. Now, here you will got the basic understandings that functionally they are antagonizing. One's function is to relax, another's function is to contract. Histamine is supposed to contract by means of H1 receptors, whereas epinephrine is supposed to relax the smooth muscle by means of beta 2 receptors. So this is called functional antagonism, being shown by the different receptors. Here we were talking about the single receptor. In case of the functional receptors, we are talking about the receptors. Functional antagonism is referring to the receptors. Different receptors, they have got different kind of functional actions on the specific site, like smooth muscles. One is contracting, histamine, another one is relaxing, epinephrine. By means of histamine is doing its action by means of H1 receptor, whereas epinephrine is showing its action by means of beta 2 receptors. That's it. Thank you.